In this video, we want to talk about the professional phagocytes. This would be specifically the neutrophils and the macrophages. Now, the macrophages are monocyte-derived cells that are found in the interstitium of a tissue, making them a first line of defense against infection. They also help in a housekeeping function by removing dead and dying cells. Neutrophils, on the other hand, are largely circulating in the bloodstream. They are drawn out of that bloodstream through a process called extravasation in response to an infection. The neutrophils outnumber the macrophages by about 10 to 1, so they dramatically enhance the response to an infection. Now, the macrophages over here on the left, in number 1, will also respond to dying cells. Dying cells express what are called damage-associated molecular patterns, right, on their cell surface. And that labels them for phagocytosis by this macrophage. In addition, we have a response to complement labeling, right, of a foreign pathogen here, in this case a bacterium, that is being labeled by these complement proteins, right? The complement proteins become activated, will stick to this bacterium, labeling it for phagocytosis. Another option is that the phagocytes will recognize, they have receptors, these pattern recognition receptors that recognize and bind to microbe-associated molecular patterns. In other words, specific characteristics of the membrane of this bacterium. And that will actually initiate phagocytosis. Finally, down here in 4, we could have antibody labeling of the bacterium, right? Another form of opsonization, similar to what the complement proteins did. But in this case, very specifically, right, as antibodies are produced from B cells in response to this particular pathogen. In all cases, these phagosomes will fuse with what's called a lysosome. Lysosomes are enzyme-containing organelles that will then digest the contents of the phagosome, right? So we form this structure called a phagolysosome, the combination of a phagosome and a lysosome. And we digest those dying cells or the pathogen into fragments. Now, in the case of the macrophage, they have MHC2 containing vesicles that will further fuse with the phagolysosome. In this case, binding those foreign antigens and presenting them on the surface of this cell to initiate activation of other immune system components, specifically the T cells. So the macrophages are antigen-presenting cells.